A good morning or afternoon to you. Thank you for joining us to learn about the promise and progress made in the development of IgM antibodies and other alternative frameworks. I'm Jan Reichert, Chief Operating Officer of the Antibody Society, and I will be your moderator. Today's webinar is designed to inform and educate attendees about the unique attributes of IgM antibody therapeutics and how these molecules can be used to address unmet needs and create new options for patients. This webinar features two expert speakers, Dr. Bill Strohl and Dr. Bruce Keyt. Dr. Strohl has been involved in biopharmaceutical research for over 35 years, and he has substantial experience in commercial antibody therapeutics development, which he gained at Merck and Janssen. He is currently president of BiStro Biotech Consulting and a member of IgM Biosciences board. He will discuss how IgM, as well as IgA and IgE, can address different targets than traditional IgG antibodies. Dr. Keyt, who is the Chief Scientific Officer of IgM Biosciences, has over 40 years of experience in the biopharmaceutical industry. He will discuss the specifics of how IgM, in particular, make exceptional antibody therapeutics. We will take questions after each talk, so please add any and all questions to the Q&A box in the viewer as you think of them. Please note, the webinar is being recorded. Without further ado, I now turn the show over to Dr. Strohl.
Hello, and thank you for joining us. I'm going to talk a little bit at a high level here about the larger, we'll call them larger antibodies, IgE, IgA, and IgM, and give you a little bit of a background so that when Bruce talks, which will be a lot more in detail, he will be able to enlighten you as to some of the new things that are going on in the field. This picture right here is an artist's rendering of IgM 2323, which is an anti-CD20 by CD3 antibody, bispecific T-cell redirecting antibody. And this antibody binds 10 sites on the B cell and only one on the T cell. And Bruce will be talking with you about how this effectively makes a very, very good T cell redirecting antibody. In broad strokes here, you can see on this slide the antibody classes. On the left-hand side, these are the circulating antibodies. And on the right-hand side, you see the secretory IgA dimer. Note that to have a secretory antibody, you have to have both the tail piece and the J chain attached. So you are only going to see secretory antibodies in their natural form from IgA dimers and from the IgM pentamers, which contain the J chain and tail pieces. We're going to talk a little bit, like I said, about the IgA, the IgE, and the IgM antibodies in this talk. So there's actually a long history, longer than probably a lot of people recognize, of IgM antibodies in clinical trials. So the very first IgM antibody in clinical trials was a rat IgM called Campath-1M in 1982, making it one of the very, very early antibodies that was in clinical trials in any format. Shortly thereafter, there were two antibodies against lipopolysaccharide, one from Zoma and the other from Centocor in the early 80s there. Both of them for sepsis, and those were very competing antibodies. Neither one of them, of course, succeeded. Just make a point, sort of a historical point, the Centocor antibody turned out to be quite significant from the standpoint that it was sort of the failure of that antibody that drove Centocor to work on what eventually became Remicade. If you look at the top here on this slide, what you see here is a series of antibodies that are all natural antibodies. They were natural IgMs from mouse, rat, or human that were then converted into therapeutics, essentially by using those hybridomas or cloning them and putting them into CHO cells. But nevertheless, they were natural antibodies. The first antibody that became sort of a recombinant, we took the variable chains from one animal and then put them into another format, is this MOV18 in 2015, which was the chimeric IgE. If you go to the next slide here, what you see is the more modern formats of IgMs, in which these are all going to be engineered IgMs, and they're either humanized or human, and on top of that, the IgM backbone itself, in most of these cases, and there are 5 here, has been modified as well. So a very different format, if you want to call it, of IgMs in the modern phase as opposed to that earlier phase. And this is where we now start to see the real value of these larger antibodies come out, where the engineering of them to make them specific for specific uses has really made some very potent and very interesting antibodies. And we'll talk about some of these. Bruce, of course, is going to talk about some of these in detail. In the next slide, essentially there are 3 reasons why one might want to go to one of these alternative isotypes. The avidity is the most obvious reason that you might think of here, in the sense that when you have 10 binding sites instead of 2 binding sites as an IgG, you change very significantly how that antibody is binding, especially if it's binding to cells where you have a multiples of receptors. And in fact, this is actually something, we'll show you some slides later on this, but this is something that's actually very significant in how the IgM actually does its work. So avidity is a very, very important thing. In the case of IgAs and IgEs, and a little bit with IgMs and maybe some other formats that are similar to IgMs, alternative mechanisms of action to kill cancer cells. So in the case of IgA and IgE, the idea would be to increase or make more potent ADCC activity utilizing different effector cells. In the case of IgMs and antibodies that are made multimeric, like hexabodies, the idea there is to increase the ability to mediate complement-based killing. The final reason, and it's gotten very interesting lately, and I'll give you a slide or two to explain this, is the access to alternative compartments. And this, of course, would be for the dimeric J-chain containing IgA and the J-chain containing IgM. Okay, I'm going to show a few slides here on the increased avidity and how that can lead to novel biology. The first example here is with the SARS-CoV-2 virus. And we actually did a study here. We made an IgM from a natural IgG. It was an IgG that came out of a library. And if you look down on the right-hand side here, you can see that the IgM is incredibly more potent against the virus in neutralization than is the IgG. The IgAs sort of form an intermediate potency. And if you look on the left-hand side, you can see an artist's rendition of this, and this is showing the IgM laid down across a series of receptors. That IgM has a total diameter of approximately 30 nanometers. So we calculated that it could actually cover as many as 4 or 5 receptors at one time. And so this potency actually allowed us to generate an IgM that went into clinical trials as an intranasal and intraoral antibody. We've since dropped that particular molecule, but it showed very, very, very good potency in vivo as well as in vitro. Another reason why avidity can be such an important factor and where IgMs can drive activity that is really not available to IgGs is with the clustering of receptors. And this slide here shows the clustering of the death receptor 5, DR5, and the ability of the IgM to cluster these receptors and increase the agonism on that receptor. Bruce will show you a little bit more about this later, but you can see here on the right-hand side with 4 different antibodies, the increase in potency over the IgG using the same variable sequences is somewhere between 5 or 6 and 12,000 fold, which is incredible. And we have an antibody, a DR5 antibody, agonist antibody, in clinical trials now that Bruce will explain a little bit more in his talk. The next series of slides, those are just 2 examples. The third example, just to make the point of where avidity can matter, is against receptors or targets for which there is very low antigenicity. So carbohydrate targets, for example, where it's very difficult to make a high affinity IgG. When you place a moderate affinity IgG antibody against a carbohydrate target into an IgM format, you increase that activity against that target incredibly due to that avidity effect. So now we're going to talk a little bit about using IgAs and IgEs to increase ADCC, and we'll just go through a few of these slides quickly. The first case here, actually, my apologies, I moved ahead a little bit too quickly here. This is another way in which increase of avidity can help to increase the activity of a particular antibody. So I'm going back a little bit now to this avidity effect, and I apologize for getting a little out of sync there. So for many, many years, people have tried to alter IgGs to make them into IgM-like antibodies. And the very early work was to take the tail piece from the mu-TP from the IgM, fuse it to the IgG, and that would result in, you can see here at the bottom, increased C1q binding and lysis of target cells. And the point here is that even as far back as 30 years ago, people were working on the ability to multimerize IgGs using IgM-based technology. In this next slide, this has been now modernized, and there are multiple ways now that people can make multimers of IgGs. And the most famous way, the most important way clinically is the hexabody, which was discovered by the scientists at GenMab. It is basically a modification of a site or two in the FC, which allowed for this multimerization effect to take place in situ. So the antibody is actually administered as a monomer IgG, and then as it sits down on the cell, it multimerizes into this hexamer or pentamer, hexamer in this case, I'm sorry, hexamer, which increases then the ability of C1q to bind and increases then C1q complement directed lysis. There are now 4 hexabodies that have been in the clinic. You can see there at the bottom, there was a DR5 mixture that has now been terminated, but it was in the clinic. A CD37 duo hexabody, a CD38 hexabody, and a CD27 HexBody, all now in clinical trials. And these are taking advantage of the fact of the multimerization, just like you would have with an IgM, the multimerization of the IgG, so that it becomes an IgM-like antibody on the surface and increase the activity of complement-mediated killing. Okay, now we're going to go to talk a little bit about the IgE and the IgA. So IgEs are important antibodies, obviously, in the whole allergic field. But there have been a couple of efforts made to use IgEs and their ability to bind the Fc epsilon R1, which is a very high affinity interaction, to increase ADCC. And I'll show you on the next slide a little bit more about that. But there is one antibody that in this particular format that has been in the clinic, MOV18, which was a folate receptor alpha antibody that was put into the clinic in 2015. Those clinical trials ended last year, and I don't know that it's continuing at all, but it was an effort to utilize this platform. It was not engineered other than the fact it was a chimeric antibody taking mouse variable sequences and putting them onto the IgE. One of the things with the IgE is that you can get, with this Fc epsilon R1, the high affinity, so you can get cross-linking that also leads to degranulation and production, secretion of cytokines, including TNF-alpha and MCP1. And the combination of these various effects leads to, at least in preclinical models and in vitro and in vivo, very, very potent antibody responses against particular targets. So there still, I think, is some interest in this antibody as an approach. But as I said, the one clinical candidate is now done. IgAs offer another way to increase ADCC, and one of the ways here is through interaction with neutrophils and through the interaction of Fc alpha R1 CD89. On the right hand side, it just shows you the pictures of the various types of IgAs. The point here is that the IgA interaction in circulation here, which would be the monomeric IgAs here at the top, these IgAs, when they bind to CD89 on neutrophils, provoke a particularly strong response, ADCC response. And you can see here on this slide that example here in A431 cells that have been transformed with HER2, that you get a much greater response here with an IgA versus the IgE with IgG with similar variable sequences. The important part here, though, is that the IgA has to cross-link that receptor. If it doesn't cross-link, it actually provides a negative response. But the positive response through the cross-linking is actually much, much stronger than the IgG type response with those cells. So because of these activities, people have been trying to make modified IgAs. And these modified IgAs include multiple efforts, two of which I'm showing here. The bottom one is including the IgA residues from the C alpha 3 replacing the CH3 of an IgG to make what they called, the authors called, an IgGA antibody that bound both Fc alpha R1, Fc gamma R1, and gamma improve both ADCC via neutrophils and ADCP via macrophages. And so this is a sort of a hybrid antibody, IgG, IgA antibody. On the right-hand side is another format of hybrid type IgG, IgA antibody, in which the entire Fc of the IgA is fused to the C terminus of the IgG. And the table at the top, which you don't need to go through in any detail, basically shows that all the activities were retained. So the wild type activities for each receptor, both the Fc alpha receptor and the gamma receptors, was retained across the board with this larger format. Again, making a potentially very potent type antibody. A final way in which IgAs have been engineered, actually there are multiple different ways, but these are the ones that I'm showing here, is the formation of a bispecific IgA using this heterodimeric format that is now quite common with IgGs by modifying the this case would be the C alpha 3 domain of the IgA, so that it would form heterodimers. And on the right-hand side, this is work done at Zymeworks. On the right-hand side simply shows their ideas or their concepts of how this could be used in multiple ways. So there's a lot of interest out there in terms of engineering IgAs, and there are multiple small companies now that have come up and are focusing on the engineering of IgAs as potential therapeutics. An important reason why IgAs also might be useful therapeutics is their ability to be secreted. And this slide, of course, shows the standard pathway for binding to the PIGR receptor on epithelial cells and translocation across those cells to the mucosal surface. Efforts have been made, actually several different kinds of efforts, and I just show one here, in which components from the IgA were added to IgG to see if you could make an IgG that could do the same thing. So on the right-hand side there, the IgG itself, of course, did not translocate. The natural IgA dimer did translocate, as you would expect. In the middle, the IgG with the tailpiece from the IgA did not translocate, but when you had the J chain, it did. Okay, so the point is that the requirement to make an IgG into an IgA-like molecule would be both the tailpiece and the J chain, as shown on the right-hand side. And so there are efforts to do that in order to make IgGs to make an IgG that has that capability to go into the mucosal compartment. This slide, which is the second to my last slide here, is a very interesting slide from the standpoint that it represents some very new work that is coming out of the Duke Cancer Center, Jose Conejo Garcia's laboratory. And there are two essential parts of this. The first was a paper published in 2021 by BiStro et al., in which they showed that mucosal IgAs, natural mucosal IgAs, provided B and T cell mediated protective humoral responses against ovarian cancer. And the reason that they were able to see this was that epithelial cancer, ovarian cancer in particular in this case, express PIGR on their surface. And then what they showed, as you can see here on the right-hand side, was they showed that you got this transcytosis, and you're able to see also the ability of the IgA to enter the cells. that led to a very, very recent report. It was at AACR, just last week, by the same laboratory, in which they showed that they could target a KRAS mutant inside of epithelial cancer cells by an IgA that was made against that target, against that particular KRAS. This was KRAS G12D mutant. And this now opens up the possibility of utilizing both the ability of IgAs to translocate to the mucosal surface and the ability of IgAs to get into epithelial-based cancer cells and potentially target intracellular targets there. This is a very exciting new set of data. It's very basic research still, but I think leads to this idea of IgAs becoming a very important potential future therapeutic. And finally, in the last slide, just showing that the use of these types of molecules, IgAs, IgEs, hexabodies, IgMs, is quite new for the most part, even though there were some very early IgMs. The fact is compared to the whole gamut of uses of various IgGs, basic IgGs, ADCs, bispecifics, high ADCC modified IgGs, silenced. Those have all been around a while, and there are several that are either in the clinic or approved. We're still not there yet with IgMs, hexabodies, IgEs, and IgAs, but you can see the potential from this presentation. You'll see more from Bruce for these types of molecules. Thank you.
Thank you very much, Bill. We do have several questions. Both of these relate to hexabodies. So the first one, you were talking about multimerization, and the question is, how does this multimerization of the hexabody IgGs work?
Well, so there are actually different ways to do it, but fundamentally what you have is mutations in the sort of CH2, CH3 region that's sort of out there on the surface that cause some affinity to those same types of antibodies. So IgG affinity. And it causes then when these antibodies settle on the cell surface for them to see each other and to come together as multimers. So there's essentially the whole idea of the hexabody came out of a natural antibody against HIV that actually showed this kind of activity, and then the team at GenMab, along with their collaborators, made mutants that made this affinity, IgG affinity, greater and caused this to happen better.
Another question. In fact, we got quite a few coming in all of a sudden, so we'll try to get to as many as possible. The next one is, why has less work been done on fusing the VH from IgG with the CH of IgM? I mean, examples of fusing 18 amino acid tail piece, etc., is more common.
Well, so historically, people have, I think, sort of putting it out there that I think people have felt that IgMs are difficult to work with, right? So there was certainly some urban legend that IgMs were difficult to produce in CHO cells, for example. And so I think, and Bruce is going to tell you that that's not the case anymore. And absolutely, it is not the case anymore. You can get multi-gram quantities of IgMs now. But I think historically there was still this attitude that IgMs were difficult to work with. And there was some attitude that they weren't that stable either, which also is urban legend because they are actually quite stable. So, I think what you're going to see now, and what part of, I think, the reason that we're having this session is to get people to think about different ways in which IgMs and IgAs also could be utilized.
Thanks very much. A couple more questions here. Do hexabodies have increased avidity over IgM? Question mark. 12 versus 10? Question mark.
So, yeah, that's a very good question. It turns out, and it depends, I guess, how you do the experiment. We've done experiments at IgM Biosciences comparing directly IgMs versus hexabodies, and the IgMs actually do appear in most cases to be more potent. But that also could be due to the fact that the IgG is forming the hexamer on the surface of the cell. Well, the other thing is that the hexamer is sort of the idealized format for these hexabodies. They actually also can form 3-mers and 4-mers and 5-mers and that sort of stuff. So you actually have a variety of different formats, I guess you could call it, on the surface of the cell. It's not all hexabody. It's a series of different mers.
Next question has to do with the biological effect. What do you think about the strength of CDC complement effect? Do you think there are indications where complement is better suited than ADCC?
That's a very good question. Hard to say. So ranking the activities in a natural environment, ADCC is the strongest, ADCP is considered next, and CDC is actually considered the third. Once you, though, move CDC to a more potent format by multimerization, then it becomes a little bit more competitive for the other two. The other thing that has been shown by the GenMab team is that as you get that multimerization on the surface of the cell, you also increase the other activities as well. So they also see increased ADCC. So it's not just a single type of activity. It's really a combination of activities.
Thank you. One more question, and I think we're going to have to move on to Bruce, though we will save some time at the end for additional questions. Last question for you, Bill, right at the moment. Is there any potential toxicity after receptor multimerization by IgM? For example, cytokine release syndrome, C1q overreaction.
So certainly C1q overreaction is a possibility. We haven't really seen it, but it is a possibility. In terms of cytokine release syndrome, it's actually the opposite. I think Bruce will be showing you some of this. It turns out that as the IgM sits flat on the cell surface, especially when you have T-cell redirected antibodies, you're seeing a little bit of a slower and more moderated, I'll call it, activity, and that results in a much lower cytokine release syndrome. And Bruce will talk about this.
Well, excellent segue to our next speaker, Dr. Bruce Keyt, who will now discuss how IgMs make exceptional antibody therapeutics. Dr. Keyt.
Okay, thank you, Janice. Thank you, Bill, for the thorough introduction. And now let's focus more on IgM. And particularly what we've accomplished here at IgM Biosciences. You see that we've put a great deal of effort into protein engineering as well as manufacturing, and you'll hear more that now we're in the clinic. And I think a key thing to remember about the early work that Bill mentioned, natural antibodies in their normal ontology in the immune system are relatively low affinity and perhaps more broad specificity. And as one of the questioners asked, we've made a great deal of progress by taking VH and VL regions of good IgGs and making them great as IgM. At least that's our expectation. There's the engineering, and I'll talk more about modification of J-chain and all the opportunities that you can achieve with these macromolecular forms of immunoglobulins. And as a result, we put a great deal of effort in manufacturing. We've got our own GMP manufacturing site, as well as we work with other teams to scale up our multiple clinical products. And so we are now well introduced into the difference of the IgG and the IgM, and importantly, the IgM hexamer versus the pentamer you see on this slide. It's really the introduction of the J chain. So we like to think of it as that IgM must have 6 members at a six-seated table, and one of which can be occupied by the J chain, which creates the pentamer. And the opportunity here is in getting clustering of antigens, whether they're free-floating antigens or cell surface antigens, but particularly in the latter case, we focused on circumstances where you can utilize that receptor clustering to drive biology. So I'm going to give 2 examples today. The first of which is the clustering of the death receptor that Bill referred to earlier. It's a natural opportunity to see this as DR5 and the TNF receptor family requires trimerization. And in this case, you see on the left, DR5 is overexpressed in many different solid tumor types and also some hematologic indications. And this has been well appreciated for years. Many teams, pharma and biotech, have taken IgG anti-DR5s into the clinic and seen some apoptosis, but we speculated that the IgM would do much better at that. And indeed, when we took 5 or 6 different programs as IgGs with their different epitopes, here you see the structures of 2 or 3 DR5s with the ligand trail 2 in turquoise in the middle. The epitopes are in red. But the important thing is, almost regardless of where the epitope for DR5 is with the corresponding IgG, the IgM is universally more potent at driving that clustering and getting the apoptotic biology in a much more potent fashion. And so that's the approach here we're going to take, and we're going into the clinic and have been in the clinic for a couple of years. But first, looking at the preclinical evidence for this. And on the right, you see a model that's often used in DR5 biology, COLO205, on the upper right panel. And here's a gastric PDX model where you get essentially cure of these mice. 6 out of 10 mice are tumor-free long after their treatment regime. I want to show the histology on the left, however, and addresses an important question many have, is that the IgM versus the IgG is too large to penetrate a solid tumor. So we evaluated this in a pharmacodynamic manner, and you see that the cleaved caspase in this histology is giving evidence that the IgM is clearly extravasating from the tumor and driving the biology of DR5. And this is a xenograft model excised from the mouse and evaluated and taken only 1 hour after intravenous administration. Now, interestingly, I don't have the slides for you, but coming back 1 week later, the IgM is still showing much stronger evidence of cleaved caspase DR5 biology. And we see that once an IgM is on site, it has a very long residence time and almost irreversible binding to a cell surface, an antigen-coated surface with the multiple high avidity valency. And so you see this in the clinic as well. Here is monotherapy treatment with our clinical candidate termed IgM 8444. That agonist activity is seen in patients pre and post treatment using cleaved caspase as a measure of that biology. So we're very enthusiastic to pursue that. And in fact, looking at this in combination therapy, you see here the results of our phase 1b study we announced in January, in combinations with FOLFIRI in colorectal carcinoma patients, seeing in this waterfall plot much better activity of the IgM anti-DR5 with the standard of care FOLFIRI. So we're looking forward to pursuing these in colorectal carcinoma. And now I'd like to shift gears to our efforts in initially in hematology oncology, treatment of lymphoma. And I'll describe that more, the opportunities of bispecific IgM CD20 in autoimmune disorders. Now, here we've altered the engineering approach to IgM, taking advantage of the J chain and its unique single integration into the IgM pentamer. We prophesied that you could add in an additional binding site to the J chain. In fact, you can do both the N-terminal and C-terminal derivatization of the J chain. This is done in a molecular biology format, such that when it's released from the cell of synthesis, we have a well-integrated IgM pentamer with a single binding site to CD3. And that was important for safety features that I'm going to describe more. So we gain the high binding power of the avidity. This gets us better targeting to low tumor. And interestingly, this results in a more controlled stimulation of the T cells, less cytokine release. So here we have some of the evidence for the high binding on these. In this slide, you see that pretty much in every case, an IgM is much more active. Now, Bill showed this in a number of examples, particularly in SARS-CoV. Here we're looking at lymphoma cells where the IgM is 100-fold more potent at binding than the comparative bispecific IgG. Where this is important is on the right. You can see even with those lymphoma cells pre-treated with an IgG, the IgM will displace that IgG. Seen in blue, the IgM gives you strong binding in spite of pre-coated cells with an IgG. This is also key in terms of driving the biology. We have two main mechanisms of action, and Bill has gone into detail on the complement binding activity of IgM on the left. Now we've introduced T-cell-directed cytotoxicity of recruiting T cells, a very potent mechanism of action where we can introduce a key specific bispecific approach using the IgM. And here you see better CDC activity, pretty much coordinate with the binding power, 100-fold higher degree of complement-dependent killing. And on the right, you see much better killing function due to TDCC, and importantly, on low-expressing cells. In this example, the Ramos cells were cultured in the presence of Rituxan, and now you see in green, they're no longer very sensitive to the ADCC component driven by Rituxan. And interestingly, the bispecific IgGs also lose a great deal of activity on the CD20 low, but the bispecific IgMs are pretty much unaffected by the reduction in CD20 20 or 30-fold. These cells have only about 3% of the original CD20. So this is a key component of using an IgM, especially against resistant and refractory lymphoma. Now, we'll take this forward again to compare these two mechanisms of action. One of the questions you heard earlier was looking at the relative potency of complement, in this case with TDCC. In red, here's the results of a primate study where we're depleting circulating B cells in cynomolgus monkey. In red, you see the potency of complement, and in blue, you see the potency of the TDCC. And in fact, in blue, you get both TDCC and CDC both mechanisms are potent, but TDCC is more so. And this also shows important features. At the very high levels, from 5 to 25 mg per kg of this antibody, you'd often find in a cynomolgus monkey with a bispecific IgG that you're into the region of high morbidity and mortality. We were able to treat these monkeys with essentially no ill effects. And this is due to a unique mechanism you see, that allows us to do a qualitatively different means of T-cell engagement. On the left in this schematic, looking at the immune synapse, you see high density connections between tumor antigens, in this case CD20, with their corresponding T cells on an adjacent, corresponding T cell receptors on an adjacent T cell. Very potent mechanism, and in fact, too potent. You get maximal cytokine release, you get maximum T cell activation. Now, the IgM is able to get that strong T cell engagement, good release of granzyme and porphyrin for cell killing, but not the maximal effect of and full cytokine release. We like to think of it as we can turn the volume dial up to 7, but not go up to 11, that you don't need and you don't want that maximal T cell directed cytokine release. And this is true, and here's an in vitro study where we look at in a human PBMC study, you get good activity of both the bispecific IgG and bispecific IgM, but the IgG bispecifics are correlated with very high cytokine release. And here you see the gamma interferon, interleukins, TNF at very high levels. But the IgM is giving you very modest to no cytokine release. And this is also true in the animal study I showed you earlier, the potency. Here we see the cytokine release in the cynomolgus monkey studies, and I'm comparing it to a series of other bispecific IgGs you see in various colors reported by other teams. And as you get to, some don't show at the 10 mg per kg because, in fact, the monkeys are often had to be euthanized at these high doses. Whereas in blue, you see the IgM bispecific is quite safe, quite tolerant in this biology. This is important for our ability to use the IgM safely in the clinic. Here you see the results in the cynomolgus monkey. We get deep depletion of the B cells in spleen in the upper left and lymph nodes in the lower right of these this quadrant of histology studies, staining both 19 and CD20. This deep depletion is going to be very important as we're looking forward to including this program with autoimmune indications where you're looking for extensive B cell depletion in order to block the B cell responses in autoimmune and, in fact, return the immune system to its more native state rather than the pathologic state. So I'll just describe some of the results we've seen recently. Here's a patient in our lymphoma trial. You see on left pre-treatment, the PET scan imaging. On the right, we're getting complete eradication here of the metastatic lymphoma. And on the right-hand panels, you see before and before and after histology, where the IgM removes the B cell component of these tumors, leaving a fibrotic residue, but it's actually having high levels of infiltrating T cells, which is what you're looking for in these bispecific treatments. So I've given you a few examples of how we've used the IgM in treatment of both solid tumor and lymphoma, and now I would like to also include in this description also our effect not only in lymphoma, but autoimmune disorders, SLE, RA. We have an active program going. We look forward to updating you soon on these. We have also initiated a trial for myeloma and a CD38 by CD3 type bispecific, and we're also headed towards the clinic on leukemia treatment with a CD123 by CD3 type of bispecific. So we find the engineered antibodies, as you see on the left graphic, can be very, can take the best opportunities of clustering receptor antigens even on low expressing cell surfaces. The CD123 will be a good example of that, but also being strategically careful in bringing in activated T cells, activating the T cells, but having a safer, more physiologic response in that T cell activation mode. So building on that, I'd like to go to the next step of engaging not just signal 1 with the CD3, but also signal 2. Here we have, I mentioned earlier that you can derivatize both the N and the C terminus of an IgM. And we look forward to being able to build an IgM bispecific with even greater enhanced potency. That will be important for driving bispecific activity in solid tumor indications, where we can get strong binding to the tumor-associated antigen, cluster T cell receptors, and further potentiate that activity with signal 2. So here, an anti-CD3 on the N terminus of the J chain and a CD28 on the C terminus of the J chain. So look forward to hearing more from our teams on this front. And finally, I would describe a series of programs that we have been able to bring in and towards the clinic. We spoke about the DR5 in solid tumor, colorectal, a broad series of solid tumors, as well as hematologic indications. Furthermore, we have imvotamab in NHL that we have continued to look at as combination therapy, but not as much in monotherapy. But the opportunity is significant in using this type of bispecific with its enhanced safety attributes, as well as the deep B-cell depletion for autoimmune disorders. And mentioned myeloma, leukemia, and furthermore, we've used this type of engineering to provide targeted payload delivery. Here on the last example, and we have a program in the clinic with an PD-L1 IgM, but we've included IL-15 on the J chain. So with that, I've given you a quick tour of the work that we've done at IgM, and looking forward to questions.
Thank you very much. We do have lots of questions, so let's dive right in. For the tumor penetrance slide, was the IgM built with the same IgG it was compared to? That is to say, the binding arms have the same affinity. This person says they're curious about the relative influence of affinity versus avidity effects on IgM penetrance relative to the IgG used.
Good question, and thank you for the question. I should have mentioned that, but I'm glad you brought it up. In all of the examples that we used in this presentation, we compare the corresponding binding regions from the IgG format and then transported that to the IgM format. So it is strictly a head-to-head comparison looking at the affinity or avidity of bivalency to the predominant avidity, in this case, of a 10 binding site. Now, as Bill had mentioned earlier, we calculate that we can get 5 or 6 of the 10 binding arms to cluster to any given cell surface, depending on the antigen and so forth. But the answer to your question is we have compared the relatively the same affinity, avidity of the format to look in these tumor penetrance examples. So you get very, very good binding and tumor penetration. It's true that we see almost irreversible binding, but nevertheless, you can see even 1 hour that the IgMs are extravasating from vessels and proceeding 5-10 cell layers deep into these tumors. And that extends over the matter of hours and days to pretty much all regions of the tumor.
Next question. Can you explain again, because perhaps you did before, what exactly causes the high cytokine release with bispecific IgGs, and how does IgM escape this effect?
Right. A very important question. And we that schematic touches on it, but let me expand a little. Is that we believe, as I think is often is well appreciated, that the MHC peptide presentation to a T cell receptor is a relatively rare event. And this has been studied greatly in tumor antigens. But in normal circumstances, it's a rare event that the T cell is exquisitely sensitive to perhaps 1, 2, 5, maybe 10 interactions, MHC presentation events to a T cell receptor. Now, via biotechnology, being able to make a bispecific antibody to such a tumor target as CD20, where there may be hundreds of thousands of CD20s on a given tumor cell, you can easily see that we've taken the natural biology of the T cell and over-exaggerated that, and to the point where we're getting too much response, too much cytokine release, complete cytokine release. And so the goal is to really return a bit closer to the natural physiology, where we consider it a less dense or not in such an exuberant T cell activation, a less dense immune synapse. I hope that's helpful in describing this biology.
Well, if the person has another question, we have a few more minutes, but we do have a lot more questions to get to. One that I really would like to know the answer to myself is, are there recycling mechanisms for IgMs? So that is to say, IgM version of FcRn recycling for IgG. How does PK compare?
Very, very good question. We have engineered IgM to participate in the recycling mechanisms, but natural IgM does not bind the Fc recycling receptor, Fc gamma receptor, the so-called brambled receptor. However, we have to remember that pretty much in all physiologic fluids, whether it's blood, synovial fluid, even CNS, the ratio of IgG and IgM is relatively the same. So IgMs do get through tissues, whether it's the other pathways, the IgM, IgA, PIGR receptors, the IgM will be cycled through the tissues. It just doesn't use the same recycling pathway. Now, we have engineered longer and longer half-life forms of IgM. I think perhaps that is also a key part of your question.
Okay, so what is typical half-life for an IgM, an engineered one?
Right, and then a typical half-life of an IgM, the textbook case of an IgM is about 5 days. That's perhaps because the IgMs in your blood have been cycled to the liver many series of events. Our IgMs are multi-day in half-life, shorter half-life than an IgG, but consistent, we find, with weekly dosing for our first program on the lymphoma, but every other week for dosing in the solid tumor indications.
Thanks very much for that. We have a couple of questions. Both of them really are touching on manufacturing. So the question is, would you please elaborate on the manufacturing challenges you have encountered, for instance, GMP-compatible IgM purification?
Excellent. Yes, and I'd have to say that's the history of IgM Biosciences. We've spent the last 10, 12 years working on that and perfecting it. And as Bill alluded to, it has been a challenge for the field at large that we feel we've largely we've accomplished. We're getting IgG-like yields now of IgM. I can say we use the standard industry standard of transfected CHO cells that produce high yield IgMs. The IgM is fully formed intracellularly and then secreted as a pentamer. And we can guarantee that if it's a pentamer, in the cases where we have engineered J chain, it is a bispecific pentamer. We don't need any extended engineering to get our form of bispecific. And we've had to modify purification techniques. We don't use the protein A immunoaffinity, but we have found a very robust series of processes that allow us to make good GMP quality IgM, and now we're working on 4 to 5 programs in the clinic, and have made this a very feasible and well-produced IgM with quantity and quality in mind.
Thanks very much for that explanation. We've got a couple of questions that both seem to be relating to off-target binding. So the question really is, what is the risk of off-target binding due to the increased avidity of the IgM format, and how do you assess and mitigate this risk?
Right. So we look very carefully at that, and I think it's a good question. It's an important question, and often initiated by all of our team's history with naturally derived IgMs. They're less affinity mature. They're indeed less specific as a result, and you can see, in a sense, that's the advantage in Mother Nature's design of the IgM to be the first reactor against many different invading microbes. Now, we differ greatly in that we take good IgG sequences that are quite specific to begin with, put them onto the IgM framework. We get the high avidity, but I can think of only one example where we've seen slight additional binding to a near-related molecule. We generally don't see any off-target binding on the IgM different than the corresponding IgG. We evaluate that with whole protein genome interactions such that we can confirm the high degree of specificity, even with the high avidity.
Okay, another couple of quick questions coming in. Is it possible to induce internalization with IgMs?
Great question. Yes, we believe that even with IgG, there's antibody-induced internalization depending on the receptor and the epitope, and so it's an additional opportunity to get intracellular internalization. And we have engineered, given a specific target and its epitope, either for low internalizing interactions or even higher internalizing, and look for further opportunities in that area. But it's a good question. We evaluate that closely.
Last 2 questions. First up, is there any immunogenic risk for the engineered IgM?
Good to take note. Now, we've looked in our clinical programs. I might start by first looking at our cynomolgus studies and compared immunogenicity of human antibodies in a primate, and largely the immunogenicity in both cases. You will get primate anti-human antibody response. It's generally to the variable domains, not to the constant regions of either the IgG or the IgM. We have found in our clinical trials that when we see very low immunogenicity, generally just in the following the first 1 or 2 doses, they're not inhibiting, they're not neutralizing, and in fact, they diminish.
All right, well, it turns out I lied. Somebody added another question that I think is worthwhile, and I think we have just like 1 more minute we can squeeze these in. How are the polyreactivity differences between the IgM versus conventional IgG and bispecific IgG?
Yes, I think that that's an important feature to look at in terms of IgM. You might expect to see greater polyreactivity, but that is still inherent to your choice of the variable domains, starting with an IgG and then collecting those variable domains and re-engineering as IgM. We don't see an enhanced polyreactivity of the IgM as long as the corresponding IgG was not polyreactive.
Good response. Last question, I promise. Given the unique biology of IgA and IgE, do you envision applying your platform to produce these kinds of antibodies?
Well, yes, indeed. Yeah, we're active on IgA, and it is a good match for our technology that we've worked considerably on J-chain modifications of J-chain and high-yield cells producing heavy, light, and J-chain antibodies. And of course, we would be remiss if we didn't include IgA. And we have examples where perhaps the best opportunity is with a decamer or a hexamer, but also a tetramer. And we get to use the different biologies that Bill referred to, the various receptors and the cell types that are most responsive, whether we're looking for NK cell mediated activity, neutrophils, whether we're looking at T cell mediated activity. So I very much appreciate that question. We look forward to much further development, and especially some of the new work that's coming out on IgAs is very exciting. And there will be opportunities to match the right target with format, whether it's an IgA or an IgM.
Excellent. Well, I believe we've come to the end of all of these splendid questions, which you answered brilliantly. So I will wrap things up here. And in concluding, I'd like to thank our speakers for providing their insights into the promise and progress made in the development of IgM antibodies and other alternative frameworks. And I thank you very much for joining the webinar today. The question was asked about the on-demand version, so I can say an on-demand version will be available soon, and a link to it will be sent to everyone who registered. Thanks again for joining us, and have a great rest of your day.
Thank you, Janice. Thank you, Bill.
Yep.
And attendees.
Thank you all.